So, as I was saying, the long axis of the calace or along the calace serves as a landmark structure or a landmark location where the chicken embryo can be seen. That the anteroposterior axis of the animal, the craniocaudal direction, okay, actually lies perpendicular to it. Now that we've established these now that we point out these important introductory concepts, let's now examine the whole mounts. Alright, so here we are seeing an early stage of the chick blastoderm. We've identified two landmark locations or structures that can be seen from the picture. Here we are seeing the uh, area pellucida, which is said to be, or which is described as a transparent area where your or our future embryo will actually lie. So this is area pellucida. Outer or external to it, we can see another structure or another region called the area opaca. And this one, right? So you guess that they are described according to how they are seen under the microscope. So the area opaca contains colored cells or I should say uh, pigmented cells, no? While the area pellucida, on the other hand, are more of transparent. So just remember that the area pellucida is the region where your embryo will actually be found in later stages. No? Also, let's take note the region identified here as P. This one is a region where the blastoderm will actually form its posterior part. No? So posterior region, caudal region of the animal is said to develop from this structure downwards. And then the whole structure here along the long axis of this region identified as P is where gastrulation of cells or where gastrulation is facilitated. No? So this is an area where gastrulation actually begins. You also take note that the cells move on the direction from the area or the more external part of area pellucida towards this structure here, this middle part. Okay, so here we can see a later stage of the chick blastoderm where uh, an important structure is now developed, the primitive streak. So this primitive streak is a region where the cells of the epiblast, the embryo proper, epiblast is the group of cells that will actually constitute more or contribute more on the development of the animal. No? This primitive streak actually elongates in the cephalic and in the caudal directions, so both uh, towards the head of the animal and towards the tail of the animal. No? As more cells enter this streak or line region, okay, line-like region, it elongates in the cephalic area initially, so more parts of the animal during this time are said to be forming majority of the or contributing to the cephalic area. No? Its cephalic extension will keep pace with the expansion of the secondary hypoblast that is actually beneath it. No? The primitive streak microscopically consists of a lightly staining groove bordered on each side by darker staining ridges. So you can see the lightly staining part here. No, looks like a canal, and then you can see the ridges that are darkly staining. So remember here, or let's introduce here an important structure called the Hensen's node. So the Hensen's node is a thickening that is formed as a closely packed cells on the cephalic end of the primitive